Shampoos used around the world. Shampoos are known as personal care products. This includes makeup, shampoo, soap, anything that you usually use on a day-to-day basis to take care of yourself. Personal care products themselves are part of emerging contaminants, which is a name for a group of pollutants that are relatively new in the aquatic world, such as medication, of course, personal care products, anything like that you use on a day-to-day basis that can get into the stream. These cannot be filtered out easily by a wastewater treatment plant, in part because they were made a long time ago. The wastewater treatment plants we have are simply not equipped to filter out these contaminants. There are several ingredients in shampoo that may have a negative effect, and such as parabens. That's a common one that people talk about today. Different shampoos have different kinds of ingredients. We all know menthol is used in dandruff shampoo, and that's what gives it its strong smell. So perhaps some of these ingredients from some of these shampoos are worse than others. My hypothesis is that one or more of the shampoos I test will have a lower LC50, so that means it's more toxic than the others. The LC50 itself is simply the concentration of shampoo and water that kills 50% of the organisms inside of it. Of course, my alternate is that they will all have similar LC50s, and I do want to point out that this is an exploratory experiment. I'm not going in here saying dandruff will have a lower LC50 than everyday shampoo. I'm just seeing what happens if I do this, which shampoos are worse for aquatic health. The shampoos I will be using include Everyday, Dandruff, Color Treated, Volumizing, Dry Hair, and Repairing. I chose these as they are some of the most common shampoos in use today, and I would use more, but that's just simply not feasible. Every single one of these shampoos have to have 6 beakers at least, plus the beakers used for replicates. And we just simply don't have enough time or resources to do a ton of shampoos at once. I will be using Daphnia dubia, the common water flea, as my test subject, mostly because these are on campus as of now. We have them in the WAEC. You feed them with selenastrum and a protein mixture called YCT, and I will personally keep them in the cool refrigerator as that's where they thrive. I will separate these specimens into 1,000 milliliter beakers, at least 30 to a concentration per shampoo, because that 30 is a good number for statistics. And then I will be exposing them to various concentrations of shampoo. And I wanted to point out that this is specifically for acute toxicity. This is not for chronic toxicity. Of course, like I said earlier, I will need a 1,000 milliliter beaker, 6 of them for every shampoo at the very least, not counting replicates. I will need my shampoos, my test subjects and their food, a pipette to feed the test subjects, a large graduated cylinder to ensure I get the right amount of shampoo per beaker, and then a stir rod to ensure the shampoo is thoroughly mixed with water before being introduced. How I will do it is I will fill up the beaker with 500 milliliters of water, and then I will add the test subjects. I will then take say I'm doing 2% shampoo, I will add 20 milliliters of shampoo to 480 milliliters of water. I will then add that to the beginning water, creating a 1,000 milliliter solution that's 2% shampoo. Here's a picture of the Daphnia dubia. They're very simple, very small organisms. And then here's just a picture of the protein mixture and selenastrum that I will be using, with the pipette, of course. This is just an example of what we could see. I will not graph it out this way, but this just gives you an example of what an LC50 test is like. Generally, organisms have some sort of tolerance. They can handle some, but then it just gets exponentially more fatal until it levels off, usually when, well, all the organisms die. I had several literatures that I reviewed. I put the most important ones up here. Not all of them are super important to the project, but I found use in all of them. The golden book for this entire thing was the EPA's Guide to Measuring Acute Toxicity. I will put that at the end in my citations. This is just a guidebook on how you do an LC50 test. I used this book for the entire experiment on how to set it up. Another piece of literature I reviewed is actually very close to what I'm doing. It's the presence and effect of pharmaceutical and personal care products in a real world situation. So this is basically my experiment done with fish, granted not with bugs, done with fish in the real world situation, measuring the effects and amounts in the real world, of course. Another one that I picked out to use was this experiment right here for two reasons. One, they used Daphnia pulex, which is very closely related to what I'm using, Daphnia dubia. So the way they treat and use their subjects are an inspiration to how I should treat and use mine. The second reason I use this is because this is also an acute toxicity test, just like my experiment. So with those two factors together, this is basically my experiment with a different chemical. The final one I thought was important enough to list here, which I was kind of iffy on this. I wouldn't really necessarily count this as a literature reviewed, but it is this experiment right here. And I just showed this experiment as it does show very well how personal care products, just as shampoo, can infiltrate a stream or a wetland or pond or any aquatic system really. And how organisms such as bugs and plants can respond to these personal care products. So to begin, before I can do the test, I have to do a range finding test. And this is just used to find approximately what concentration I should be looking at. This looks at 50% shampoo versus 5% shampoo. And whatever, say, in these example concentrations down here, say I ended up at 5%, then I wouldn't need to go up to 10% or down to 1. I would just do, say, 5%, 4%, 3%, all of that. Which is what I show right here. So let's assume that 5% was the closest to the true LC50. A lot of bugs died here, but a lot of them lived too. Then we pick at least, emphasis on least, 5 values, not including the control, clustered around 5. So my values would probably be right here. This will be a non-renewed and static test. I did both of these because this is the simplest LC50 test you can do, and I figure the more I mess with it, the greater chance that I have to mess it up. So non-renewed means I just put the water and the shampoo in there and leave it alone, other than feeding. I don't put more shampoo in, I don't do water change, nothing. And static means like there is no water flowing. I'm not putting it into an artificial stream, it's just a beaker. This will be done over a period of 48 hours because that is the longest you can go without cleaning. And I figure if I clean the beaker and I accidentally stress them out or kill them, that's going to mess with the results. And of course, you always should do replicates with this kind of test. And the next figure I'll show you, they actually do the replicates really well. And here's that figure. Notice that they do have 10 organisms in each vessel, but they have 3 concentrations. So this is 30 organisms per concentration. And this is just their examples of concentrations used. This is a good way to set up mine. The results, I will be measuring concentration in parts per billion because that is what you see most often in the field. If you take a water sample, say at Ritter Park, you'll probably find shampoo parabens in parts per billion or caffeine in parts per billion, which is just 0.001 milligrams a liter. And what I'm specifically measuring is the number of dead in each beaker. And this will be, say I have some breeding and I get 32 organisms in each beaker, I can also do percent dead if I absolutely need to. I'm also going to be recording the no observed effect concentration. This is just the concentration at which I could not see any increased mortality. I'm not going to actually do anything with this at all, but this is just a nice piece of information to have for a future research project. So for interpretation, we have a few options, but the only one I can realistically feasibly do is graphical. This is the worst method because say my LC50 was closer to 5 but had some killed on 2, partial mortalities you can't work graphically. There has to be an all or nothing basically. For studies that have partial mortalities, it's recommended you use one of these. These are by far the better choice, but I personally do not understand them at all. They're way above my head, and that's why I will be sticking to graphical interpretation. Graphical interpretation basically consists of connecting the two concentrations bracketing your mortality LC50 with a line and then plot them against the base log 10 of percent effluent. I'll show you what that means. So here are your concentrations, and here, so what I mean by partial mortality is your lower concentrations, there were no deaths. All deaths occurred at these two concentrations right here. This is the only one where there is any mortality. And when you have something like this, then you can bracket this highest one with no mortality with this lowest one with 100% mortality to find your LC50. This is a very bad method, I will admit, but it's the only one I am capable of doing. Possible source of error, of course, always include human error. I could miss a feeding day. I could accidentally make a 3% solution instead of a 2% solution. I could stress them out, anything like that. Another possible source of error is not enough randomization. Randomization comes in randomizing which individual goes into which beaker, of course through stratified random sampling, and measuring like where in the refrigerator the beaker will be placed because maybe it's colder in the back and warmer in the front. So you need to randomize where your beakers are going to be. Another source of error would be getting some old specimens in there. You're supposed to use very young neonate specimens so they don't die off naturally. But as someone who is not experienced in this, I could very well accidentally put in a near death water flea that would be able to skew the results. For further research, I think it would be nice to isolate which ingredients, say dandruff shampoo and volumizing shampoo, both have a really high mortality rate. Then I can look for an ingredient that both of them have that none of the other shampoos have, and perhaps that's why it's super high. So if these specific ingredients are especially toxic, we should study them more. And then I think I would expand past invertebrates. I'm really interested to see what the effect on algae would be. And of course, everyone always wants to know what's going to kill the fish. So these are two possible ways to further the research. And then here's just my works cited. Thank you very much and have a nice day.